Okay, ladies and gentlemen, today we are going to discuss viruses. And as you can see up on the board, we have two different pages in our interactive notebook. One's dealing with the shapes of viruses, and the other one's dealing with the parts of viruses. So definitely, when we're thinking about some objectives for this lesson today, number one is definitely to know the different shapes of viruses and the names of the different shapes. Second, you need to know the different parts of the viruses and their functions. So, how we are going to start is we're going to discuss viruses, a little bit of background information. Then we're going to go to the shapes of the viruses and finally finish with the parts. So here we go. Viruses are not living. Okay? Viruses are not living. We wouldn't classify a virus into any one of the domains. All right? Now, when I look at this, we might ask, okay, why? Why do we not consider viruses living? Well, number one, they cannot reproduce on their own. They need to have a host cell to reproduce. If you would go way, way, way back into our first discussions in biology, we talked about characteristics of living things. So if we could grab your interactive notebook A and talk about characteristics of living things, I can go through because I know I've got my one up here somewhere. All right. So when I look, okay, and this is the beauty of it, okay, characteristics of living things. Page 37. So, wow. Remember doing this little flip diagram here? Seth, I know you don't remember doing it. But, share similar chemicals. Okay? Here's the one where I put the zebra. I almost called it a giraffe. No, I know that's a zebra. A zebra in the little baby zebra reproduce. Characteristics of living things right there. They can reproduce. So, by that statement right there, that viruses cannot reproduce on their own, that is one reason that they are not a living thing. However, viruses do reproduce. They just need something to do it for them. They do not react to stimuli. That was another characteristic of life. They react to stimuli. Viruses do not. They do not react to stimuli. They are not made of cells. Viruses are not made of cells. They don't grow and develop. It's not looking, oh, look at that teeny little cute virus. He's going to grow into a big virus someday. No, doesn't happen like that. They do not grow and develop. As you're going to see when we talk about tomorrow the life cycle of a virus, okay, once they leave that host cell, that's as big as they're ever going to be. So they do not grow and they do not develop. Viruses, however, do mutate. Viruses do mutate. Now, we talked about different types of mutations earlier on. Well, viruses have genetic material. So that genetic material can mutate. We talked about different types of mutations: deletions, insertions, substitutions. Okay? And those were all dealing with a genetic code. Well, viruses contain genetic material. So viruses will mutate. Okay? Viruses will mutate. There's certain drugs that the virus, okay, let's say there's a virus. It used to be a really, really effective drug fighting this virus. All of a sudden, it doesn't work anymore. Why? Why? That the virus mutated. Therefore, that drug doesn't affect it anymore. So then the drug makers have to come up with a new drug to fight off that virus. Now, shapes of viruses. Shapes of viruses. The very first one, this virus happens to be the influenza. You've probably had the flu before, haven't you? You've probably had the flu before at one time or another. The influenza virus, that virus is what we call an envelope. An envelope. And it has different, see these little, they call them an N spike and an H spike in this diagram here. That these spikes are in the envelope. The envelope's this little yellow circular thing, okay? We can see different RNA segments in here. And these little purple dots are the capsids. Now, right here, these different spikes, and we're not going to go into this in depth, but have you ever heard of the H1N1 virus? Okay? That was not too long ago, wasn't it? H1N1. Those are just the H spikes and the N spikes, and they number them. So that H1N1 means different numbers of the H and the N's. But like I said, we're not going to go into that very deep as other than just what we did right there. Okay? So that shape is an envelope. My next shape, you can see the RNA right there, and you can see the blue capsid that is surrounding the RNA, made out of each individual protein subunits. That right there is helical. That is helical. The third one we're going to talk about is I don't even know how to pronounce that. What do you think? Icosahedra? Sound good? Icosahedral? That's what we'll go with. Icosahedral. Notice the little green capsids. This is pointing right here to that green spot, but it's pointing inside. Inside those green capsids is the DNA that goes with this virus. Okay? And different spikes on the virus. Now notice when we talk about genetic material, right? When we talk about genetic material, we know the difference between DNA and RNA, don't we? And we've already seen different viruses that had DNA. This one, the one right before, the helical, that had RNA, correct? So it could be RNA on the virus, it could be DNA on the virus. Okay. Way. And when I taught AP biology, we even talked about retroviruses. Okay? Different types of retroviruses. Now, when we talked early on, we talked about the central dogma, didn't we? Anybody remember what the central dogma was? Watson and Crick? Okay. What was it? What was the central dogma? Come on. Look in your notes. Okay? Look in your notebook. In your notebook somewhere, you should have stuff about the central dogma. Is it in your table of contents? Is the central dogma in your table of contents? It should be. We're close. Did you find it? I found it. Right there it is, isn't it, Jamie? Okay. What is the central dogma? I think I said 50. The central dogma. Jamie's is on page 54. See that? DNA to RNA to protein, the central dogma. So the DNA codes for the RNA then translates the proteins. Well, some viruses work backwards on that, we've found. Some viruses take RNA and make DNA. All right, so they take an RNA script and make DNA from the RNA. So that's a different type of virus. And if you want to take AP Bio later on in your high school career, you probably could learn about that. Now, my last shape is the guy that you have on the opposite page there. Okay? This is what we call a bacteriophage. A bacteriophage. And you can see that there are different things that go with the bacteriophage. So right here, the top. Okay, the top, and you're going to have to identify these in a little bit here, so look up here and pay attention. That is what you have labeled in there, the capsid. Okay? All the little squiggly stuff inside the capsid is the DNA. You don't have to label the neck or the collar here, but look at this thing. This is the sheath. Okay? Yours is called a tail sheath. So we have a tail sheath right there. A base plate. You'll learn here in a second the function of that base plate. And finally, we have tail fibers. So those are the different parts of this bacteriophage that you're going to have to label here coming up in a bit. Now let's talk about these parts of a virus. We're going to start off with, up. You'll see this again on your page right here. I'll leave this up later on, okay, because this is the last slide also. But on your virus, you will have to label these in these specific colors. Okay, so here's your color codes for coloring the virus. All right, but I'll have that up. That's my last slide too, so I'll show you that here in a second. Let's talk about the capsid. The capsid, first of all, if you would lift your parts of the virus up, you have notes under there. And your notes start with a protein shell or coat that surrounds the genetic material. So, these capsids are made of proteins. They're made of proteins. They're kind of like a protective coat. They protect the DNA. Okay? It protects the core of the virus and allows the virus to infect new cells. So the whole function of the capsid, the proteins that surround the genetic material, is to protect the virus. So as you can see, here's just yet another view. Okay, here is the capsid. And it's also sometimes referred to as the head of the virus. Now, does a virus really have a head? No. But we just, it's up at the top part of the virus, like our head's at the top of our body. So it's just an analogy. The genetic material. We talked about the genetic material of the virus. Okay. We said, first of all, it could be what? It could be DNA or RNA. So it could consist of deoxyribonucleic acid, or it could consist of ribonucleic acid. It could be single-stranded DNA, or double-stranded DNA, single-stranded RNA. All right? It cannot reproduce without a host cell. So viruses do not have the ability to reproduce their DNA and their RNA. They need another cell to do that. So inside the head of this bacteriophage is the genetic material. The tail sheath, that middle part, the tail sheath, it contracts like a syringe. Well, what happens when it contracts like a syringe? It forces the genetic material of the virus to travel from the capsid through the sheath down to where the base plate has connected to the host cell's membrane. Then it travels through the host cell's cell membrane into the host cell. And we'll talk about that in a little bit here, okay? In a little bit here. Actually, we'll talk about that tomorrow. So you'll understand that whole process of the tail sheath tomorrow a little bit better, because we're going to show you this, okay? I'm going to show you the whole idea of the virus connecting to a host cell and show you the whole life cycle that happens with that. But that's tomorrow, okay? Just believe me that the function of this tail sheath is to act like a syringe and to force the genetic material into the host cell. Okay? Here's that sheath. So basically what happens is the genetic material from up here travels down through the sheath and is forced out of the base plate into the host cell. The base plate. The base plate is where the virus attaches to the host cell. Okay? The base plate is where the virus attaches to the host cell with the help of the tail fiber. The tail fiber look like the little legs of the bacteriophage. So the tail fibers are the little legs. It fixes to the receptor site of the host cell and allows for the reproduction of the bacteriophage. Okay? So, If it doesn't have something to attach to the host cell so that they can place the bacteriophage's DNA or RNA into the host cell, it's not going to work. So this base plate is what attaches to the host cell. The final part we're going to talk about here, and you can see the base plate there. I got it in yellow in this little diagram here. Final part we're going to talk about of a virus is the tail fiber. The tail fiber. What happens is the tail fibers help attach the base plate to the host cell. Okay? Kind of like they hold on to the host cell so that the base plate can connect through the cell membrane. Okay? So they help attach the bacteriophage to the host cell. Now, I told you I would come back here with this next one. Okay? With this next one. And here it is. I'm going to leave this up on the board for you to color your parts of a virus. All right? So we're going to have to color the parts of the virus. And also, I have another sheet here, and it's a virus dichotomous key in which you are going to you're going to have to cut and color these different shapes of the viruses here. Okay, so I'll pass this out, but I'm going to leave this up on the board for you to do your actually coloring of the parts of the virus.